Hello, everybody, and thank you all for being here. I am Mauro Petretta, Senior Scientific Advisor at RegenHU, and I am thrilled today to be opening this masterclass on fresh 3D bioprinting together with Dr. Andrew Lee, Head of Bioprinting and co-founder at FluidForm. Hi, Andrew.
Hi, Mauro.
So, hi. I will start with a presentation on our side on the basic principle of bioprinting and why the fresh method is so relevant right now in the field, and then I will give the floor to Andrew for a more detailed explanation of his working principle. Allow me to share my screen. And here we are. As I said, let's start with the basic, that is the very definition of bioprinting. I'm confident most of you already know it, but for those who don't, well, several versions of the definition have been provided over the years, but they are all based on the same concept, that is the ability to dispense simultaneously living cells and biomaterials with accurate spatial control in order to fabricate three-dimensional structures. Due to its unique features, bioprinting has gained a great relevance in the tissue engineering field, and it's a proper revolution. For example, being a computer-aided technique with an accuracy down to the micrometric level, it greatly increased the complexity of the structure we are able now to fabricate, while at the same time guaranteeing the ability to have a standardized and repeatable product. That was not the case with the previous fabrication technique. Moreover, it enables the possibility to follow personalized and patient-specific approaches for different applications in the biomedical field. Due to its unique features, bioprinting has gained rapidly relevance in many different applications of the biotech field, starting from the production of engineered tissue, where it can mimic the natural one through a standardized, reproducible, and automated 3D process. Also, recently, there is a great interest in developing through 3D printing and bioprinting technique, novel tablet formulation where it is possible to tune the release profiles and combine different active compounds in a single tablet to prepare patient-specific formulations. And last but not least, one of the field most widely explored in recent times through the bioprinting approach is the fabrication of tissues and disease models that can be used for drug discovery projects or for personalized diagnostic and advanced therapeutical treatments. One of the key factors to consider when preparing a bioprinting process is the choice of the right biomaterial or bioink. Bioprinting has in fact some unique features, but it also comes with some very specific characteristics, like the need for the cells to survive to the dispensing process. For this reason, for example, it's not possible to apply high pressure levels to avoid excess shear stress to the cells. That's why the most commonly used materials in bioprinting are one with a relatively low viscosity, but with a quick gelation or cross-linking mechanism after deposition. I try to categorize them divide them in three categories: thermosensitive materials, which exhibit a tunable sol-gel transition upon temperature and pH change, such as, for example, collagen. Photoactivated materials, where usually a functional end group is grafted into an existing polymer, so that in presence of an initiator and a light source, the cross-linking can happen, such as in the case of GELMA, just to cite a very used one. And also, I tried to refer in a very broad sense to chemical for all those mechanisms in which the gelation or cross-linking is achieved by exposing a gel precursor to different initiators or to complementary reactive solutions, such as in the case of alginate and fibrinogen. Of course, these materials come also with challenges, especially in the field of 3D printability, that is certainly dependent from the material properties, but not only. It's also tightly related to the dispensing technology, the chosen process parameters and cross-linking mechanism, and of course to the complexity of the architectures you want to design. And these factors combined together can make the difference between the desired outcome and the unsuccessful achieved one, as you can see in the picture. And it is to target these and many other issues that, gathering the feedback we received over our 14 years of experience from our user community, we developed our latest platform, the RegenHU. The goal is to provide the means to fully control and optimize the process by means of our software solution, like our design and process management software Shaper, and our controller interface to monitor your process and adapt on the fly your parameters to correct it immediately. And our hardware solution, our RegenHU platform, that enables to dispense a wide range of material by combining different dispensing technology in a unique and personalized configuration. Starting from the hardware side, as I said, each configuration can be unique and personalized according to the application and can equip a wide range of different printing techniques such as pneumatic or volumetric extrusion, drop-on-demand technology, or high-temperature melt dispensing to process a wide range of possible biomaterials. But this is not the only thing that is required for an optimized bioprinting process. For this reason, also auxiliary tools to provide different cross-linking mechanisms are available in terms of, for example, thermal control of the work zone and the printeds, or the possibility to include light curing kit to cross-link photoactivated material with different wavelengths. The goal is, of course, to follow the users through all the steps of the process, from the preparation and design of the experiment and the project to the actual printing protocol and the real-time monitoring of the process and evaluation of the results. But no matter how optimized your process can be, there can still be limitation related to the material properties of the complexity of the architectures due to the nature itself of the bioprinting process. For this reason, several approaches have been tried by researchers over the years. The first one involved the use of the support materials in a multi-tool bioprinting approach. And though it is possible to achieve a really wonderful result, has been proven multiple times by researchers exploiting this technique, this approach anyway presents some limitation concerning the maximum achievable geometric complexity or size, limitation in the material selection and the coupling of support material and functional material, and of course a slightly increased process time due to the fact that both the support material and the functional material have to be printed. And it is for this reason that free-form fabrication techniques have lately emerged as an innovation in the field, because they tackle all these limitations, enabling by printing your material directly in a support gel bath, as Andrew will explain us in detail in a few minutes, to print structure with unlimited geometric complexity and can be scaled to organ size, while at the same time also enabling the possibility to provide specific functionalization while the printing process takes place. Now I would like to show you my experience with the fresh method. I selected the blood vessel network as a model to print. Through our Shaper software, I defined all the parameters related to the internal architecture, the toolpaths and the dispensing tools that I would like to use. Moreover, through our software, it's possible to select to perform the process directly within multiple well-painted or Petri dish formats selected from a library. Finally, through our sequencer, it is possible to visualize the toolpath in a layer-by-layer fashion and to define dispensing, but also non-dispensing action, such as, for example, calibrating, cleaning, or curing. Let's move to the print itself. Here we can see the printing process take place in the support gel kindly provided by FluidForm. For this application, we selected our volumetric strand dispenser printed in order to have an accurate control over the flow rate, and the template silicon-based material, just to highlight the capability of the technique. You can now see the blue structure taking shape within our support matrix. And let's move to the end of it to see the final outcome, which is our blood vessel network printed and removed from the support gel and ready for your research and testing phase. This will be all from my introduction. I thank you all for your attention. I would like to give a special thank to our user community. It's thanks to their constant effort that we also keep learning and evolving every day. And of course, I thank you again for your time, and I invite you, if you have any question, to address them during our next Q&A session or to contact us through our email or social channels. Now I would like to give the floor to Dr. Andrew Lee. Hi, Andrew.
Thanks for the introduction and the awesome presentation, Mauro. Let me share my screen as well. Give me 1 second. And remember, in chat, you can type down the questions and we'll have time to answer at the end of this presentation.
Exactly. If you want to address us any question, feel free to write them in the chat, and then we will, in the end, take a dedicated time to answer them.
All right. Well, thank you, everyone, for coming to this webinar. And thank you, RegenHU, for hosting us. Today, I really want to talk to you all about, thinking about how bioprinting and tissue engineering as a field could be looked through a pretty different lens, because we think we at FluidForm have the technology platform that's available for creating very complex prints. And I want to talk to you about why. So really thinking about bioprinting differently, why that's important, how do we grow as a field all together to move forward towards our goals of creating something that is functionally relevant to a clinical setting, and then again pushing a path towards clinical relevant research is what we're aiming towards. And so just a little bit about myself, I finished my PhD in 2019 at Carnegie Mellon University in Professor Adam Feinberg's lab. My thesis was focused on really exploring the use of bioprinting for engineering complex architectures found in muscle tissues. And so this leveraged some of our work that led to the development of FRESH, which I'll talk about. I was fortunate enough to work on a very collaborative team of researchers, and we were able to publish a lot of this work in the journal Science in 2019. So in this presentation, I will share with you a lot of the images and data from this work. It's a couple years back. And since this is really the core technology that we at FluidForm is founded upon, and is really conveys our vision of how we propelling bioprinting towards becoming key to regenerative medicine, tissue engineering, and really biomedical research as a whole. So, at FluidForm, we want to be the first to print organs. And of course, that is a very, very ambitious goal. And we recognize that. And in order to get there, there are several steps that we want to take in order to get there. And we really see that, bioprinting can be boiled down to two key problems that must be solved for us to progress in biofabrication. First, I think many of you on this call are working on, these materials are unique in their desired properties, right? These could be some sort of electroactive material. This could be a tunable stiffness material, something that you've developed in the lab or found interesting to work with in your system. However, most of these materials actually turn out to be very difficult to print and assemble in 3 dimensions. So as a result, a second problem also emerges. So it's not possible then to fabricate the desired geometry that you might want. And so this is using the materials that you want to print with. And so really, we at FluidForm have developed the technology to make it possible now to print these materials that you might desire to print in any geometry. So I like to show here just a print that we've done in the past. This is a scaled down model of a human heart that's a model derived from MRI and printed entirely out of collagen type 1. And, shown here, I know most of you, this looks pretty familiar to most of you. On the left is FDM printing of thermoplastics, and on the right is, SLS printing of metals. And, for a long time now, a lot of people have asked, could we leverage these very well-established processes in additive manufacturing for the 3D bioprinting of biological materials. And, the challenge is something that we've discussed earlier, is that these biomaterials that we might want to print with are oftentimes very soft, right? So we're talking about a lot of these very familiar biomaterials that are used in tissue engineering, such as your cells, collagens, PEG, T-cell ECM, alginates. These are all very soft materials. And one benefit that plastic and metal printers take for granted is that the materials that they print with are a lot of times far harder than the soft water-laden hydrogels that make up our body, where plastics and metals can be heated and then cool down very quickly to form into stable shapes. These protein hydrogels can't do that, right? These protein hydrogels that make up your body, they can't do that. So one analogy that we'd like to make is this watch assembling problem. So if I were to give you this disassembled Swiss watch, you'll probably look at this and feel that it's very complex, but you can probably rebuild this, because even though these parts are small, they're all very rigid parts. And with some time, some knowledge, some YouTubing, and some practice, you can most likely learn to put everything back together despite the very complex arrangement of parts. However, if you imagine all of these parts are now soft, they become very hard to assemble, very hard to put together. And when they're intertangled with each other as well, this level of complexity drastically increases. And you kind of end up with these clocks as seen here in this very famous Salvador Dali painting, where everything slops and droops everywhere. So you can see this in this video here. Here we're just printing pure collagen type 1 into a dish in air. And you see that this is just meant to be a very a normal rectangular slab with sharp edges and a decent height. But you'll find that when printed, everything kind of coalesces together. You'll get rounded edges. You don't get defined features, and you lose resolution. And that's not something that we want, right? And the same phenomenon happens with each of these soft materials, and each of these materials come with their own unique printing challenges that's related to their rheological properties, their gelation mechanisms, and the gelation kinetics. So this is really the first core problem that makes bioprinting so difficult, right? And so what we've done as a field is, look at each of these materials individually. This is what we've done so far, and assess what makes them difficult to print, right? And modify the materials for their printability to make them printable. So, you can imagine starting with this kind of hard to print soft biomaterial, as I've shown before. What we've done previously is that we can modify the material chemistry so that we are tuning its gelation mechanism and kinetics so that it's gelling quickly upon deposition onto a petri dish. We can modify material composition to really tune its rheological properties by, say, add in a rheological modifier so that you can allow the material to stand on its own and be stackable layer by layer. Another way of dealing with this soft material printing problem is that you can print additional scaffolding, hard scaffolding material that provides structural support for your softer materials. And each of these do come with challenges. When you modify the material chemistry, you oftentimes reduce the cellular binding domains, increase hydrogel stiffnesses, lose functionality in your material that you've modified chemically. When you've mixed in different rheological modifiers, you oftentimes lower the concentrations of every component that's involved in your mixed material, and sometimes you don't want that. And then when you have these stiff structural elements, you need to figure out how to deal with them. You need to remove them, or maybe you're comfortable accepting that they will remain in your print throughout the duration of your study. And so, this then leads to our second problem. Because we need to so finely tune each material so that they become printable, we usually don't have quite the perfect printable material. So we're usually at some in-between point where materials are somewhat printable, but they need to be printed in simpler geometries designed around the challenges of printing them, right? So what we kind of end up with are, shapes and geometries that are very recognizable to us, but are really ones that are simpler in their geometry and ones that we aren't too excited about, right? As researchers, we want to tackle the complex geometries, the complex functionalities of these prints. And to summarize these two core problems that we see to bioprinting, again, you have these unique materials that you want to print but are hard to print. You have desired geometries that you simply cannot print with these materials that you have. And you might have noticed that we haven't even begun to consider the functional properties of these constructs that we're printing. So as engineers, usually our design process starts with outlining the functional criteria for the thing that we're trying to create, and then picking the right materials and the right geometries to best create that thing. But currently, we think that bioprinting and the bioprinting process is kind of reversed. We'd spend a lot of time making a material printable, finding a geometry that is printable, and then figuring out what that combination leads to in terms of the function of the constructs that we create. And so frankly, we want to change that thought process. So before we kind of dive into how we do that, I kind of want to invite you on this brief journey, a thought experiment. That's kind of fun, I think. If you were to imagine all of these problems that we discussed previously didn't exist, and you can print anything that you possibly want, what would you print? So what would you print if you were able to use the right materials in the desired geometries and explore the printed functions in depth? So for me, I might imagine starting to look at some of the capillary scale structures, how that branch out into arterial networks and to organ scale level networks, and perhaps even study the entire vasculature system and how perfusion happens and why it matters in a kidney, for example. Bone is another example of different scales of architecture and pattern that's very interesting and has unique functions that we can begin to replicate and mimic potentially for our implantation purposes. And another thing, what about a composite of multiple tissue types? So in this example, looking at the hand, really starting to understand how muscle, tendon, cartilage, and bone organizes together to provide the function in relation to each other and as a whole organ as a whole. That's something that is interesting to me personally. An interesting one to think about is you could potentially create devices that interact with soft tissues around them. And these devices need to have very specific interface geometry as well as its functional geometry that needs to be created. And these are very important challenges to tackle. And for the more science fiction addicted researchers in us, we are, particularly drawn to designs like these that don't exist in nature but look biomimetic and have potential, for example, more efficient fluid dynamics as an example of the functions that we can create in these geometries. So these are really the number of scientific questions that we want to study that were previously difficult to study without 3D printing. And so we think we've really developed the technology to allow us to start the print at this level of complexity and to actually ask these scientific questions. So Fresh is the technology platform that allows us to print with soft materials in any geometry for function. And Fresh stands for free-form, reversible, embedding, and suspended hydrogels. It's really a kind of printing known as embedded 3D printing that many of you might have heard of and are familiar with at this point. We no longer are 3D printing in air, but inside of support material that cushions our soft ink while it gels in place. So it's the same layer by layer process that occurs in FDM printing, only it happens inside of the support material. So in this image shown here, you'll find that we have a needle stuck inside the support bath. So this is our syringe containing a soft material bioink. We have our extrusion needle, of course, and it's extending into the support material that's printing these letters FluidForm. And the support material is shown here in yellow. And the support material is really where the magic happens, right? It has a unique property. It is a yield stress material, meaning that it will flow like a liquid with application of a force. This is very similar to how peanut butter, how toothpaste works. You can, for example, in this case, you can push the toothpaste out of the tube onto your Bristol-Cert toothbrush, lay it down, and when you flip it around, it'll actually stay there because you're not applying sufficient force to allow it to flow. And so, this is made possible because our support material is made of these small gelatin microparticles that together contributes to this yield stress behavior as a slurry. So this process of fresh printing is really pretty simple, intuitive when you watch it happen. So in blue here, inside the syringe, inside the glass syringe, we have a alginate, just dyed blue. And then we're printing into a support material containing calcium ions, which allows the alginate to cross-link the moment it gets extruded out of the needle and hits the support bath. So you'll see that this is the same layer by layer process where we're depositing this gelling fluid bioink, which is alginate, into the sacrificial support material. The bath is able to yield around the needle as it's traveling through the support bath. And then at the end of the print, you'll find that because the support material is made of gelatin, you can raise the temperature and melt away the support material to release your print at 37 degrees Celsius. And this is really what allows us to print per function, print with any gelling material and print in any geometry. So, for these next several slides, I want to show you some images and data from our work in the science paper and highlight what is possible to be printed using fresh. So in 2015, we initially described fresh in Science Advances, followed that up in the publication in Science, and then this is what we've licensed at FluidForm to really tackle our own internal challenges and really think about how we can help the community adopt this process so that we can together work towards the same research goals, which is developing clinically relevant therapeutics. So first, I want to show you this idea of printing with multiple materials that have orthogonal gelation mechanisms, meaning that they have distinct mechanisms that don't interact with each other. So if you imagine you have 4 different materials, say you have a collagen, a methacrylated hyaluronic acid, a fibrinogen, and an alginate. Each of those have distinct gelation mechanisms. Collagen will gel when neutralized, since it starts out as an acidic solution most commonly. Methacrylated HA will cross-link in the presence of light exposure. Fibrinogen is enzymatic, driven by cleavage by thrombin. And then alginate, as we discussed earlier, will cross-link in the presence of calcium ions. So if you think about our support material, which kind of looks like this, with a lot of gelatin microparticles, surrounded by a fluid phase that you can really modify and change. And this fluid phase, you can change the pH neutral buffer, you can expose the support material to UV light, and you can add calcium ions and thrombin into the support material at the same time. So shown here, we have a single printed on a single printer, printing 4 different materials that the colors of which correspond to the colors of the letter. So you'll see that it's pretty easy to do 4 materials in a single print. And in terms of thinking about the basic building block features that we can print using fresh, we really started out by taking collagen type 1 as an example to explore this, because collagen is, as we all know, is the most abundant protein in the human body and really has such a large footprint in biomedical research. To understand that, in order to understand its role in biology and disease, it really allows us to leverage that understanding for therapeutics. So here we are printing collagen type 1 in different sized filaments. So we start out with 200 all the way down to 20 micron filament diameters. We can very easily pack these filaments laterally, stack them, space them out, and then assemble them in 3 dimensions. And so taking these printing capabilities, we wanted to see how well this holds up in a large print over multiple lane scales, from the micron scale to the centimeter scale. So we chose the heart as a proof of concept model, given that at the time Adam's lab was particularly interested in cardiac tissue engineering. And so, started out with an STL model that's derived from an MRI file of a human heart, scaled it down printed this heart model. It's roughly the size of a small strawberry. So shown here in the center here, this is the actual print, printed entirely out of collagen type 1. If we take a cross-section of that print, we can see the internal details where you see some of the trabecular structure of the heart, and also some of the high-resolution infill structures that you can find here. We were able to take a micro CT scan where in these different view planes, you can find, again, different features such as the right and left ventricles, trabeculate again, the infill. There's also valves complete with their leaflets, and then also the chamber walls that provide the structural integrity of the entire print itself. And the heart, of course, was really just a scaffold, right? It doesn't really have any particular function. It was a proof of concept showing that we can take printing from the small scale to a large scale with relative ease. And so, we set out to really think about how can we recreate function in some of the key components of the heart, with the first one being the valve. So we took a model of the valve STL file, and we printed that entirely out of collagen type 1. You can see here, these are different views of the valve printed out of collagen. We were able to handle this printed valve, show that the leaflets are patent and that they are able to be manipulated and opened and closed, as seen in this video here. And then we can also put this valve into a pulsatile flow loop and show that the leaflets open and close. Of course, this is all very preliminary data still. If we're trying to think about tackling how do we create patient-specific valves that are easily implantable with the right function and able to either grow with the patients as they grow or remain in the body and integrate it with the human body. This is something that we're actively working on to tackle as well. And so the next critical piece to the puzzle of engineering heart tissue, or tissue, any tissue for that matter, is that any tissue requires a dense network of vasculature, right? That supplies blood and oxygen and nutrients to its cells. So one of the key challenges in tissue engineering has been the ability to fabricate vasculature network in 3 dimensions with control over the organization and geometry of these vessels. So we took a computationally generated approach where we took MRI data, and then we were able to create a network of vasculature based on with different sizes. We isolated just a small piece of that in order to just show proof of concept of being able to print different length scales of vasculature. So shown here, this is a see-through model where you can see some of the larger vessels extending down into some of the smaller vessels. And we took that and simply printed that out of collagen. We were able to take this print, which looks very much like our originally intended design. We took this print and optically cleared the print so that it's easier to see the perfusion. So we immersed it in a BAB solution that some of you might be familiar with, so that we can actually see the perfusion of just a simple blood mimic glycerol water solution perfused through some of the vessels that are printed here. So these vessels extend from a couple millimeters down to a couple hundred microns, which we think is pretty impressive. And then, so the next steps with this is how do we start to integrate prints of the scale with tissues that need to be kept alive. And of course, a very key functional component, perhaps the most well-known of the heart, is its contractile activity that allows the heart to pump blood. And this is orchestrated by highly organized heart cells, cardiomyocytes. And so here we created a very simple model of the left ventricle, which is responsible for pumping oxygenated blood throughout your body. So a common model of the left ventricle is this half of a football shape, American football, with a open lumen in the center and just a shell around it. So shown here is actually an actual printed ventricle model. We were able to take a cross section of that. So this is looking top down at a wall of that shell, right? So we printed this ventricle model out of 2 materials, collagen and cardiomyocytes. So you can find collagen in magenta here. printed in 2 layers surrounding the cardiomyocytes that are printed in the central layer. If we take a closer look at that central layer, we find that cardiomyocytes are very nicely elongated and aligned and also well interconnected. They're aligned in the direction of the print, circumferentially around the ventricle model, and they're very densely populating the central region where we printed them. When we take a look at calcium imaging of these ventricles, really using calcium imaging as a proxy for visualizing the contractility of our cardiomyocytes that have been printed, we find that they're electrically active. They form a syncytium that allows the propagation of a uniform calcium wave across the entire surface of the ventricle. Looking top down, we find that the same can be said, where we have this unidirectional propagation of the calcium wavefront. We are able to take this ventricle model and actually point stimulate it, allowing us to measure our conduction velocity of our ventricle model. And again, this is really just, I think this is really exciting for us. This is really just a start of being able to leverage FRESH in order to create in vitro models that Mauro talked about for the purpose of cardiotoxicity testing and efficacy testing in the pipeline of drug development. And this has implications across all different fields of drug discovery. Anything that is ingested in the human body could potentially cause cardiotoxic effects. And if we're able to create a complex model that allows us to predict those effects cheaply and effectively, then that is something that could be very impactful. And so we've really seen that others in the field have also really taken on this shift in mindset for bioprinting and adopt FRESH as a key piece of their research. We've seen FRESH grow rapidly in peer-reviewed publications, highlighted as in some of these papers shown here in just the last 2 years. And really, I just want to show here some of the other example prints that we printed, a different version of a heart valve that we've created, manipulated here, a ACL anterior cruciate ligament model that you can see we're kind of bending and twisting around here. This is our intern who had the privilege of playing around with these on a afternoon. So you can see that these are very easily manipulable. And then also a trachea that has been printed, and you can find its open lumen, some of the features on the outside of the trachea, as well as being able to close that lumen and allow it to reopen. And we also printed this intervertebral disc model, where this is really leveraging the idea of, can we use different infill structures to create different mechanical properties? And I think that's pretty self-explanatory. If you have lower material infill, you will end up with something that's softer in that region. And so you can find that this is mimetic of what you might find in an intervertebral disc, where that central region is really just this jelly-like material, and the outer region is very fibrous and provides structural stability of the disc itself. And so, FluidForm as a company has taken on this mission of really expanding the reach of FRESH, because we truly believe it can have significant and lasting impact on biomedical research. Our support material is commercially known as life support, which is really simply a sterile powder that you rehydrate into the support material that you previously saw, and you can go from sterile powder to printing within 15 to 20 minutes. And what I want to emphasize here is that FRESH is really simple to adopt, right, and slot into bioprinting research workflows. As I mentioned throughout this talk, FRESH now makes most biomaterials printable. This allows you to create almost any design and any geometry that you can think of, or a file that you might have that you can modify for printing. And this allows you to print on any printer, any hardware. And RegenHU's printer is a very well-suited platform to do that. And then if you have a accompanying ecosystem of software that allows you to communicate with that hardware, that makes it all the much easier to work with, and to really start thinking about what materials you want to print and what designs you want to print. And so this is why we've really partnered with the RegenHU to really think about how can we make all of this very easily adapted for that system, and what is the best way to make the customer experience most positive. And so, I just want to challenge you at the end of this talk to think about what will you print. If you don't have any constraints, if you now can print with any materials and really can expand your geometry to almost unlimited geometries, what will you print? And I want to leave my email address here, as well as some of the contact infos on social media. And feel free to reach out to myself or Mauro at RegenHU for any questions. And I think at this time, it would be appropriate to really open up with any questions.
Thank you, Andrew, for the wonderful presentation. And now I will say we open the floor for discussion. I see there are already some questions in the chat. We can start to address them. Well, for the optimized video option, sorry, there was not, or at least that I am aware of. Fatima, for what concerns change the pressure while printing, it is certainly possible with our Argent platform. The pressure, as well as any other parameters in your printing process, can be changed through our controller software in real time at any point during the pressure. And we know why that is useful, because it allows to save time and cut costs by optimizing your process on the fly and not being required to stop and start every time. Then I see another one for me. What is the minimum thickness you can print? Well, it of course depends on the chosen technology and needle size. Usually extrusion printing technology go in the range of, let's say, tens of millimeters, depends on the needle you select for this. While, for example, in our platform, by the integration of electrospinning and writing technology through a convergent approach, it is possible also to have dispensing of micrometric or submicrometric fibers in the same process. And so there is a discussion ongoing on the example you provided, Andrew, but you clearly, in my opinion, explained it very well during the presentation. So the print you performed were not done with a RegenHU printer, because one of the good things of your method is it is compatible with any kind of hardware and printer, so it's kind of universal, but still having an optimized system in terms of software and hardware can provide a really helpful way to exploit it to the best, or at least that's my opinion.
And this was something that we worked on, 2 years ago, with technology that was 3 to 5 years old. And that was, I really wouldn't, it's a pretty rough time sometimes to convert your own plastic printer into a bioprinter. And without, sometimes you just don't have the time to really mess around with the hardware and software conversion. It's just much easier to have an integrated ecosystem.
Yes. Then I see some other discussion about our printer. Yeah, probably you have all generation of our instrument, Gisela and Fatima. In the Argent platform, is now possible, exactly why we listen to all of your feedbacks, to change all the parameters, including the pressure, in real time while you are printing. I can assure you of that.
So I think in the questions, there are several questions.
Yeah, I let you start in answering this one.
I think I see Hatton. Is there a platform bio inks to be used for a majority of cells? I think that really depends on, that's going to be my answer for a lot of these types of questions. I think it really depends on the application that you are going for. For example, when we printed the ventricle model, we used a fibrinogen matrix to temporarily hold the cells together. We didn't really care what was the bio ink. We just wanted the cells to stay together while we were printing. And then once printed, you're able to release the print and be able to culture them in media. So it really depends on, you need to consider all aspects of your system. What do you want the cells to do while they're inside the syringe? What do you want them to do once it's finished printing? And do you want something that is in there that's interacting with the cells? So I think that's something that for you to think about, but also feel free to reach out if you have any more questions on that.
Okay.
I see that there's a question from Milkilwa. Is it possible for me to get training on the whole process of bioprinting from the beginning to the final product? Mauro, I will give you that question.
Sure. Yes, absolutely. So we, of course, are really invested in divulgating and diffusing the bioprinting potentiality all over the community. So absolutely feel free to contact us. We will plan together a way to introduce you to the bioprinting process and our platform. And we have, of course, dedicated experts, both from the technological and the application and scientific point of view, that will be more than glad to answer all your questions. So contact us through our email or social channel, and we can go further in that discussion. Then Manuela, when we talk about bioinks, yes, we usually refer to living cells suspended and embedded in hydrogel for the purpose of bioprinting. How long can they survive depends from a whole series of factors. Let's say the cell, the material in which you embed them, and most generally speaking, the condition at which you keep them. So, for example, their life could be enhanced if you keep, through the temperature control options of your printer, the printed at 37 degrees, or if you perform the printing process already in a heated medium at 37 degrees, or in a multi-wall field with culture medium. Depends really on the application and what you want to achieve. As for the survival, let's say, time, as I say, depends from all these parameters, but let's say as an indication in a range of an hour or more or less so, depends on how you treat them. Then there is a question for you, Andrew, from Leon.
Yeah, so what's the best way to create gelatin nanoparticles, and what concentration? How do you evaluate the nanoparticles? So I would say nanoparticles are hard to create. Well, not hard, but they will be hard to process and maintain. It's some of the process that we've outlined in the science paper that you can take a look at. Might be challenging to reproduce, and so that's why we would recommend using just a support material, the life support that we offer, as just to save you time, so you don't have to worry about making the microparticles themselves. But you're more than welcome to take a look at the science paper in the materials and methods section and see if that's something that you can replicate. I think there's another question for me. Is there a strategy to select the optimum formulation for support material and bioink to combine them for fresh printing and adapt the process to new bioinks? Yeah, of course. I think that starts with understanding your bioink that you're trying to create. For example, say you're really you start with something that's simple as alginate. You know that alginate needs calcium ions in order to gel. So you want to be able to print the alginate out of the syringe, and you also want it to gel inside the support material. And so you know that the support material is made of gelatin microparticles, so you can very easily resuspend those gelatin microparticles with a calcium chloride solution. So that allows you to leave the alginate ungelled inside the syringe, extrude it, and the moment it hits the support bath, it will gel. And so this will apply for any bioink combination that you might have, right? So really thinking about what is the gelation mechanism that you have, and is that compatible with the support material, which is made of gelatin microparticles. So this is life support. Another question for me, it looks like, from Peach. Can you please let me know how to print out a small artery, which can resist 30 to 40 millimeters mercury burst pressure. What kinds of bio ink we use to achieve this goal? That is a hard question. I think that is something that a lot of people are trying to tackle. A good place to start with is collagen, I would imagine, right? You have two challenges here. You have the challenge of printing the artery small with the resolution that you need, as well as just reproducibility of your printing method. And you need to, once printed, you need to have the print survive burst pressure, which is a pretty complex force if you think about it. Your print needs to really survive tensile stress as it's as pressure is being applied from the inside. And so I think that takes an understanding of the material properties of your bio inks and what is its properties after it gels, which is, I think, something for you to explore. But happy to talk more about that as well.
Absolutely. Feel free to contact us. Maybe tell us what you already tried, and we can go in the discussion a bit further on the topic. That's for sure.
Yeah.
Another question from Peach. Our network was quite large. There is a way to print out a capillary-like system for an organ. Well, it depends, of course, on the model and the geometry you design. So there can be let's say the resolution of the printer, it's below the micron, and the repeatability is in the range of few microns. So of course there is a way to reach that in terms of technology. Then what you need to work is on, let's say, two different things. One is the source model for your capillary-like system. Either it's designed or imported in some way, but it can go much smaller than what I've shown. And the other, of course, is the also answering the question, also referring to the question we answered below, and that is also in this tab below. It is possible to print smaller structure. It depends on the minimum needle and filament size, and it is strongly dependent from the technology. So you can go much lower than what I've shown with smaller needles, as long as your material allows to be dispensing them, and your system provides enough pressure to do so. And as I said, the minimum needle size can be in the order of tens of microns for extrusion, and down to the submicrometric scale for electrospinning or writing technology with our system.
I think there's a combination of different questions in chat and also the questions. There's what is typical concentrations of cells you can achieve. This also, again, depends on how you mix up the inks that you create. You can go from as low as 1 million cells per ml, even less. And for us, we go up to quite a high concentration, about 300 million cells per ml. So, and then also I've seen some questions with, have you grown organoid that contains cells? I'm assuming this is also, have you grown that organoid containing cells? I'm assuming this is also asking about, have you printed organoids? For us, we haven't printed organoids, but that's something that you can load organoids into a syringe and print with. So, yeah.
Yeah, for what regards the regulatory expectation, it is also, this is quite a complex field, because bioprinting is a very new technology, and it is, let's say, all in the making. So regulation is still evolving to adapt to the bioprinting field, and it is also strongly dependent from the country in which you are trying to use your products.
Sure.
And you have a supplier in Europe, Andrew?
Yeah, I think just email me or Mauro, and then we can give you that information. And then we do give out samples, on a pretty limited basis, but we do give out some samples of life support, just to make sure that you can try and get started with. It's something that obviously most people haven't used before. So.
And then let's move to the other chat. And So can fresh printing be combined with coaxial printing technique for printing living tissues? I see no limitation in that direction. So using a coaxial or coaxial printer together with fresh method, and then of course, as in any case, when you print vasculature or some, let's say, empty vessel, you will have to get rid of the support material that's also inside the structure and not only on the outside. But that's the only thing I can see. Correct me if I'm wrong, Andrew.
Yeah. Yeah, I think this is the coaxial. Yeah, there's no challenge to really. It's the creation of the coaxial needle that is the challenge, and making sure that it prints consistently out of the needle. So I don't see any issues with that. Eric had a question for how long can cells survive in inks that are deposited inside the life support material, kept at a temperature that keeps life support as a yield stress gel material? It's a good question. Depends on your ink, and also depends on what you suspend the life support in. So typically when we're printing cells, we would mix the life support with the media that the cells generally needs. So some sort of growth medium or some sort of extra supplemental medium that allows the deposited cells to be in a neutral pH while they're just deposited inside the life support. And at some point after you're done printing, you would release the print and swap out that life support, that melted life support, with actual full medium, so that you're now just culturing your printed constructs inside your cell culture medium. So that's typically how we print cells, and we found that it works pretty well for printing between 2 to 4 hours. So it's worked pretty well for us in the past.
Yep.
And John had a question for John, what is the most effective method to sterilize support bath material for cell-based printing? The good news is life support is sterile to begin with. So really all you need to do is work with it inside a biosafety cabinet, keep all your reagents that interact with the tube sterile, and the whole thing will remain sterile.
Yeah. Back to the question, Cliva. You are absolutely welcome, and I take the opportunity of your question to also remind everybody that is interested to know more about the bioprinting world that they can contact us through our email info@regenhu.com or through our social channels, and or through our website, and they can book a specific session with us to learn more about the bioprinting world, to see our products in action, and to learn more about specific aspect of the bioprinting technique.
Mauro, can you type your contact info in chat? I think there's a couple of questions asking for it.
Sure.
I think Akash had a question. What humidity would you say is ideal to maintain a droplet of cells in medium for about an hour for them to attach before filling up with medium? It depends on how much medium you have. A drop of cells in medium for about an hour for them to attach before filling up with medium. Typically I think your construct would dry up if it's just covered. 10 microliters is pretty small. I would say typical incubator humidity should be sufficient, which is, I believe it's close to 90% plus. So I would try the incubator, like a cell culture incubator first. You can always get a humidity chamber and try to raise that humidity to make sure it doesn't dry up. Something that you can experiment with. Yeah, print in the printer and then transfer it to the incubator, but make sure you cover it is my suggestion. Make sure you just put a lid on it that you might have for the container. Or perhaps get a better seal by parafilming it, and then you should be okay. And then in chat, I think that's most of I think we were able to answer all the questions here, Mauro.
Apparently so. Let's give a few seconds for people to eventually come up with new ones.
Sure.
But I think...
Yeah, thanks for everyone for coming. Really appreciate the questions as well.
Oh, okay. So the printer question, I think it was already addressed before, as Andrew explained. The method is compatible with any printer, and their previous work, I think it was made on a custom-made instrument. But the good thing of the fresh method is its openness to all the platform, and of course having a better platform will result in a better process. So having something that allows you to control the printing parameters and your design by means of software and hardware, and to process as wide a range of biomaterial as possible will certainly make a difference.
Yeah. And then, is it possible to reuse life support? It is not possible to reuse it, because once melted, you wouldn't be able to recreate you wouldn't be able to get back those shells and microparticles. So it is a consumable, and you typically will use about, it really depends on your print, you need enough life support to cover the entire volume of your print for your container as well. So
share the email address. You can contact us for any info about product demonstration or getting more info about the bioprinting world.
great.
And then I think that will be all.
Yeah.
Thank you again, Andrew, for your time and for the wonderful presentation. It was a pleasure to take part in this masterclass with you.
Thank you, Mauro.
And thank to everybody for being here and for your time. I hope you had a wonderful experience and could get some interesting information from this webinar, from this masterclass. And please contact us if you have any more in-depth question, any curiosity, or anything you want to learn or know about the bioprinting world and the fresh method. And I wish you all a nice evening or day according to your time zones. Bye.
Bye, everyone.